The title of my conference lecture, as you can see there, is Ecology, Evolution, and Cancer, Lessons Learned, especially through the prism of a non-ecologist, non-evolutionary biologist, but as a medical oncologist. I've taken care of men with prostate cancer for over 30 years and run a lab looking into the science of why prostate cancer cells grow and spread throughout the body. And originally, I had a title here that I basically said, thank you to you all as a field for helping me be a better physician. And I want to tell you a few lessons I've learned and a few stories along the way about how my thinking about cancer has changed because of you all and what you do and the folks that I've met over the years. As far as I'm concerned, we're here because metastatic cancer remains incurable. It's the second leading cause of death globally. It's going to kill 10 million people in 2019. About 1 in 6 deaths is due to cancer. You need to absorb that fact, okay, that metastatic cancer, despite everything we're doing, and except in a few very specific cases where immunotherapy has worked or a disease like testicular cancer, we're not curing cancer. And we're not curing it with conventional thinking. And why I keep trying to meet new people and come to conferences like this is exactly this reason. We're not winning at all. So the first lesson that I wanted to just say to you is that I've learned that cancer is an invasive species in the body. Now you may say, well, that's obvious because it exactly patterns tumor growth in the primary organ, surviving to transport to other places, metastasizing, and then there's this period where the cancer cells don't grow and establishes a new area in population biology and ecology that's called a lag period. In metastasis biology, we call that dormancy. And then there's eventual growth, and in the case of an invasive species, an ecological impact and organ destruction. So I have to tell you that I didn't know that until probably less than a decade ago. Because as a cancer biologist, cancer equals crab. So I would think about it as crab. But we, as doctors, think about cancer as something we can't defeat. It's so unbeatable that in my mind, it was an apex predator, not an invasive species. It was something that was going to attack and was undefeatable. So just that simple change for me to think about all of a sudden that this is actually billions of cancer cells interacting in the body, and not a few cancer cells, but billions, and that they were spreading just like invasive species does, was a huge lesson for us as clinicians and to understand. The reason I'm pushing this with you all is as you talk to clinicians and other scientists who aren't in your field, who aren't in the field of ecology or evolutionary dynamics, they may not get something this simple. And for us to make progress, we have to teach each other language and learn each other's language and terminology. And I was hung up on this. And so I wanted to open here by saying this was one of the first lessons I learned. And it also taught me a sort of new ways to think about the idea of cancer cells spreading that I worked for years in the field of dormancy, and I never thought about the fact of how invasive species work and that there's a lag period and we can potentially target and learn from that. So that was lesson number one. Lesson number two, I've simply titled Swamp Gas. So these are patients that I've autopsied who with prostate cancer. I had the privilege of running a rapid autopsy program so we could harvest tissue from men with cancer. And I learned a lot about how cancer spreads. And one of the things that we learned is, I autopsied 80 of my own patients, and I didn't know how they died. It's not how cancer kills kills patients, kills people is not clear. It's not, and in fact, if you ask most medical oncologists, they hem and haw. They actually don't know how cancer kills people, and actually most patients actually want to know. And I point out that this liver cancer this cancer filled with liver here, or liver filled with cancer here, cancer filled with liver, liver filled, this is the cancer, this is the normal liver. This patient had normal liver function. You can wipe out 90% of a liver and have normal function. This is a bone marrow full of prostate cancer nodules. This patient had normal bone marrow function. It takes a lot to wipe out normal function. So how do we explain this? Well, we've turned, as Keller mentioned, to thinking about tumors as ecosystems and thinking about cancer cells existing as species in a complex habitat with other host cells. In the field of medical oncology, this is a pretty controversial idea, or was at least a decade ago. But the idea that these cells can interact with all the normal body cells creates, in their habitat, creates an ecosystem, and that this ecosystem in turn, the ecosystem of the cancer, exists in the larger patient host biosphere. So we, as an ecosystem here in Cambridge or Hinxton, are in an ecosystem, but it exists on the biosphere of Earth. And we have to think about cancer, not only what it's doing locally to an organ, but what it's doing to the whole patient as the host. And because of that, we started to think about, again, learning from ecology. We learned about niche construction and allogenic engineers like beavers that modify an environment, or autogenic engineers that modify environments by changing themselves like a tree. And what we really know is that if you look, cancer is both an allogenic and autogenic engineer. It modifies the environment around it. And we started to think about that as it's creating its own swamp. And that as it changed that local environment, it was going to attract new host species of cells like tumor-associated macrophages and cancer-associated fibroblasts to that environment. But it's very clear also that cancer exists no homeostasis. By definition, it means uncontrolled growth. If you look in the dictionary, that's the definition. And that many things happen when you have uncontrolled growth, including lack of nutrients, causing changes in the cancer cells themselves, like having them undergo an epithelial to mesenchymal transition, and you have more uncontrolled growth. But I want to emphasize that this does not mechanically destroy the host biosphere. It does not wipe out an organ, except very rarely. So without that feedback loop, you get a healthy pond evolving into a swamp. And Sarah Abbott, who's here, is going to be talking to you about some ideas we have about swamp and eutrophication and how that causes changes in cancer itself. But what do cancer patients die from? Well, about 40% of patients die of cachexia. And about 30% die of a thrombotic syndrome, like a blood clot to the lung. About 20% actually die because they have uncontrolled pain and end up taking a lot of morphine and then not eating and falling asleep. And the common denominator of all those syndromes is not a mechanical obstruction. It's cytokine overproduction. So if you think about cytokine overproduction, you really are talking about a miasma of cancer. Miasma is a highly unpleasant or unhealthy vapor, and comes from the ancient Greek term meaning pollution. So it's a disease, in Greek, it's a disease caused by a foul environment. So the majority of cytokines that kill a patient are actually produced by the patient themselves in reaction to the cancer. If you think about the total number of cells in the human body, about 100 trillion, at death, We know that a kilogram of tumor is about 1 to 2 trillion cells, and that is incompatible with life in general. If you have a kilo of tumor spread out in your body, that somehow is killing you. So it's not about the cancer cell mass, it's about the context and what it's doing to the human patient. We actually studied this a decade ago, and by looking at patients who were dying and had bad cancer, we found that there was a common set of cytokines and chemokines that are basically overexpressed by all cancer patients, including IL-1, IL-6, TGF-beta, CXCL12, et cetera. And if you look at where that comes from, IL-6, one of the most evil cytokines in the cancer world, it's produced by macrophages, T cells, muscle cells, osteoblasts, fat cells, and only very little by cancer cells. It's actually the fact that the cancer cells are interacting with that human host that is caught, and they're reacting to the cancer, is producing that cytokine. TNF-alpha was originally described as connexin. It's not produced very much by cancer cells. It's produced by immune cells, and fibroblasts and endothelial cells, all in reaction to the cancer. MMPs, matrix metalloproteinases, that remodel the tumor microenvironment, the major source of them is tumor-associated macrophages, not cancer cells. But tumor-associated macrophages don't come to the tumor microenvironment until they're called there, attracted there by cancer cells. So those cytokines and chemokines basically cause a syndrome called cachexia that leads to fat breakdown, muscle breakdown, lack of appetite from those cells getting into the brain and telling it that it's not hungry. It's a vicious cycle that I'm, again, not going to spend time describing today, but it's these common cytokines, IL-1, IL-6, IL-11, interferon gamma, TNF-alpha. Same thing, those same ones in some patients interact with the endothelium to cause a blood clot in what's famously called Virchow's triad of hypercoagulation, stasis, and endothelial cell damage. And in some cases, those clots break off, go to the lungs, and cause a pulmonary embolus and death in a lot of patients. But this is why your patients with cancer get blood clots. So the summary of part 2 here is that what we call the cancer swamp, or the miasma of cancer, poisons the patient. And it's very important to think about that in terms of not just the cancer tumor microenvironment and the ecology of that primary or metastatic tumor, but what it's doing to the whole biosphere of the patient. Cancer, lesson number 3, and I'm sure we'll hear more about this at this conference, is that adaptive therapy works. Adaptive therapy has been recently written up by Jessica Cunningham, and is also been championed by Bob Gattenby at the Moffitt, and I know Carlo's very interested in. Are you going to talk about adaptive therapy at all here? No. So Carlo's very interested. Huh? Oh, Sandy Anderson will talk about adaptive therapy. So we're going to hear more about this, but one of the quick lesson that I want to leave you with is that every time you treat a cancer with chemotherapy or hormone therapy, so anytime you give a systemic treatment, you can't cure the patient, but you put it into temporary remission, but when it comes back more vicious. It comes back more resistant, it's growing faster, and in this set of my patients from almost 20 years ago, what we saw was the doubling time is measured by PSA for us, which is a blood biomarker. In hormone-naive cancer, the cancer was doubling about every 35, 36 weeks to a year. But once the hormone stopped working and you were castrate resistant, that dropped down to 20 weeks. And every time we treated with therapy, it got worse. So one of the things that I've been doing over the years is treating the way we treat metastatic prostate cancer at first is to give a castration therapy with, or what we call hormonal therapy, because we don't want to say to the guys we're castrating them. We call it hormonal therapy. It takes their testosterone down to castrate levels, and we induce menopause in the men. And that's very effective, as you can see from the black line as measured by the PSA. Basically, it goes down to 0, stays there for an average of about 2 years, and then starts to rise again, and that's called a recurrence in the onset of castrate resistance. One of the things that we've done is instead of using this castration therapy, we use hormone therapy light, or what's called peripheral blockade, where we leave a guy with a circulating testosterone, but actually block the testosterone from hitting the cancer cells. It's just 2 pills a day. We put patients on that for 3 months, and their PSAs drop, and then we stop the therapy. And we then wait until the PSA starts to rise again to about 50% of where we started, and then we treat again and keep doing that. And as you can see, each of these patients represents, or each of these lines represents a different patient. We've been able to avoid castration therapy in many men for many months by using this sort of adaptive approach. And I, quite frankly, was doing this just out of instinct and not really measuring things very well until the Moffitt group really really codified this with therapies that start, and when they start here, the adaptive therapies that they're using start here. So we can talk more about that, but I just wanted to set the stage for those talks in that adaptive therapy works. So in the last 30 minutes, what I want to show you is a bunch of new data that has changed the way I think about cancer and the evolution of cancer, and basically make a plea with you to help me understand this better. And this starts with the story that I told you. Cancer when it's metastasized, is resistant to all known natural and synthetic drugs or essentially any combination. Normal cells don't have this capability, but why, how, and when does that happen? So this is where lesson number 4 is, basically I should tell you, I'm confused. I'm confused about the concept of tumor cell heterogeneity and how you explain mutations and evolution in the concept of tumor cell heterogeneity, and then what we know. So this is the famous life family tree that's been published multiple places, multiple times. And I was trying to figure out, where is cancer on this tree? So if we're way over here as humans and we have a single cell that mutates, as we think about how, Charles Swanton and others have published these dendrograms of how mutations happen. And the problem with these is it makes you think there's only a few mutations. But cancer is made up of billions of cells that are very heterogeneous. In fact, if you take this liver patient, there are more cancer cells here than there are people living on Earth right now. So but we're all the same species, but we all look different. So how much of what's going on here is phenotype versus genotype versus true evolution versus, I've convinced myself I don't know what to call cancer anymore. And in fact, if you look back, there's been obviously a lot of literature about, cancer turning on embryonic genes, turning on archaea genes. But to me now, cancer looks like this, not this. But I don't know what's important. What I do know is that there's very few true driver mutations. That this tree doesn't explain why cancer is resistant. It just is a way of marking mutations that tell us a cancer changed over time. So that simplicity puts us thinking in a box that is totally inappropriate. And in fact, if you look for mutations, I spent 10 years doing this. I did exome sequencing and regular, all kinds of sequencing looking for mutations, when the idea everybody thought that the driver mutations were going to be the answer. But if you look at, here's again from the autopsy series, you look at the pattern of mutations. First of all, there is no pattern. But if you look at that, you can't get any hint about metastasis and no hint about drug resistance. So how does that work? How are we going to explain cancer in evolutionary terms? For a while there, I thought I knew, and now I don't anymore. And in fact, at every level there's tumor cell heterogeneity. We looked at a bunch of masses on a patient who had a rapid autopsies, a bunch of different ones. And what we saw when we just looked at phosphoproteins is that there is no pattern. And they're all over the place. And in fact, within the same lesion, you get heterogeneity. Between two lesions, you get heterogeneity. And between different patients, you get heterogeneity. Heterogeneity of the mutations is overwhelming. So so I've had to step back now, and one of the things I'm hoping to do here at the conference is try to get some perspective back, and I hope we have some time to talk about that. But that gene sequencing trip that we took down that road, publishing in good journals, taught me that we can't. Dr. Smithers' famous quote from 1962 said, "You can't learn much about what causes a traffic jam by studying the fine structure of the internal combustion engine, and genes are the fine structure. And it doesn't tell you anything about the person walking in front of your car and you slamming on the brakes and causing an accident. It doesn't tell you about your texting. It doesn't talk about epigenetics. It doesn't do anything, except we have to know the fine structure, and that's what we're studying. That's my worry. How do we put the evolution into the ecological context is, I think, the challenge that we're facing today, because it's not a linear problem. So as I said, all the cells are resistant. Where does that resistance come from? You can't find it on sequencing. So the prevailing theory has been that there are billions of cancer cells in a tumor, and by sheer stochastic chance, some cells exist that have resistant mechanism, like drug pumps, that will repopulate a treated tumor. No winnable strategy can be developed to defeat this tumor cell heterogeneity. Okay, that's the prevailing theory. I should know I wrote it in 1989 as part of a review. That's what I thought. And if you look at the NIH, what it was funding back in the 70s and 80s, it was all about drug resistance. That's what the majority of R01s was on. Now nobody talks about drug resistance anymore, because we said we knew we failed, so we went to omics to find driver mutations that we could target. That didn't work, so now we're doing immunotherapy. And nobody talks about why cancer cells are resistant to all known drugs anymore. So to help us study this, Bob Austin and his talented grad student Casey Lin and my talented postdoc Gonzalo Torga put together over the space of a couple years an engineered cancer microenvironment so we could start studying interaction of cancer cells with host cells, as well as establish stresses to understand how cancer cells reacted to that stress. And what we really were thinking about, or what was really important about this particular microfluidics device, is that we were able to create a gradient. And most microfluidic, when you do cell culture, there's no gradient. You put stuff in there, and it's not, there's no gradient. Everything just gets exposed. And we wanted to study how a gradient of stress, whether it was chemotherapy, change in nutrients, different cytokines, low oxygen, would change how cancer cells behaved. And as part of that, what we did was we were also, as Carlo mentioned, we were studying game theory. So we wanted to think about how cancer cells that were, say, epithelial and cancer cells that were mesenchymal would interact with each other. So what we did in this first experiment was we basically said we're going to put in green PC3 cells, which are mesenchymal in characteristic. So PC3 cells are a prostate cancer cell line that everybody uses. And we also had RFP labeled or red PC3 cells that were epithelial. So we subcloned out an epithelial mesenchymal group of cells that were stable, and we wanted to see how they were interacting. And the idea was we were going to put high-dose chemotherapy, which is docetaxel here, at a dose that should kill 99% of the cells. And there'd be zero, and we said, maybe the mesenchymal cells are the ones that are resistant and the epithelial ones aren't. Let's see if we can learn about drug resistance. And what you can see here is this is an experiment over 17 days, and it's been looping. But what I want to show you is right here at the end of about day 14, we got the growth of resistant cancer cells. Within 2 weeks, cells that should never have been there were there. And it wasn't just the mesenchymal cells that did it. If you look down here, there's a group of epithelial cells that did it. These, for me as a medical oncologist, this was one of the scariest things I'd ever seen. Because we treat a patient, and 3 weeks later we treat them again with the same drug, and then we treat them again 3 weeks later. We treat them with cocktails intermittently. But I saw the emergence of drug resistance with my own eyes for the first time because I had a gradient and I had a movie. You couldn't do this without a gradient and a movie because you're just looking at cell culture. So we published the device, and we published some of the game dynamics around this. But what I want to show you now in higher power is where everything took a left turn for us and have taken us down a new way of thinking. So now what I'm going to do is I'm going to blow up this box. I'm going to show you what happens for this resistance to emerge. So what you'll see as it comes back into focus is you'll see all these cells, and you'll see them clear out because the chemotherapy is working. And then sort of midway through, you start to see the emergence of these very large, very fast, big cells, giant cells. So we said, well, what's that? They're living in 10 nanomolar chemotherapy. They shouldn't be there, and yet they're very much alive, and they're moving around. And then, towards the end, you see a bunch of tiny cells sort of expand out clonally and fill the space. So now what we've got is two unexplained phenomenon. One, we saw resistance, and we saw these giant cells, and then we saw these little cells, also resistant. Totally unexplainable to us, and very, very weird. So in summary, the first thing we found was these large polyploid multinucleated cells that were highly motile. And to just show you a punchline, we thought, okay, well, how come nobody's talked about these? I mean, we can't be, I've been doing, prostate cancer, putting on therapeutics for 30 years. I never bothered with these. Well, it turns out that there are 74 papers in the literature going back over the last 30 years describing multinucleated polyploid cells in response to chemotherapy, radiation, low oxygen, changes in the tumor microenvironment. The trick is that basically 72 out of 74 of those papers say they don't survive, that they're trash, that they die due to mitotic catastrophe, that they're senescent, that they may be associated with chemotherapy, but more associated that your chemotherapy is working. And it's got nothing, that very few people ever thought that these were functionally important cells. So they reported their association, but not that they were causal. So Sarah Ammond, who you'll hear from, and basically just wrote a review about these, and we went back in the literature, and really it turns out that Mueller in 1938 or 1838, the first person to really describe cancer cells under the microscope, talked about, he showed multinucleated cells. Virchow in 1858, when he described cells, actually showed giant cells and multinucleated cells, describing them as various polymorphous cancer cells, some of them he thought degenerating, some with multiplication of nuclei, very big. But again, static, fixed cells, static, not dynamic. In fact, the very first cancer cell line, the famous HeLa cells, isolated by George and Margaret Gay at Hopkins in 1951, you can look at their original description of HeLa cells, and you see these giant multinucleated polyploid cells. So what we believe, because we had the movie and saw this dynamic change, is we hypothesized and thought that maybe these PGCCs, these polyploid giant cancer cells, are actually central to tumorigenesis, metastasis, and actually resistance may not be because they're of tumor cell heterogeneity, but really that these, since we saw them first in the movie, maybe they're the important cells. And what have we discovered so far? Well, it turns out that if you look at any tissue culture flask, and I've challenged all my colleagues as I've traveled around to talk about this, we think this is true, nobody's told us otherwise. Look at any tissue culture flask right now, and you'll see that these cells, these giant cells, are anywhere from 1 to 10% of the population, generally around 3%. And that, we said, okay, so it's not just PC3 cells, which we saw in the movie, it's all cells. And it's not, and then we said, well, maybe it's because we're using a mitotic poison like docetaxel, and we're inducing these because we're not letting them divide. Well, it turns out you can use any chemotherapy, radiation therapy, or hormone therapy and do this. It's any stress. It's just a matter of stoichiometry of how many cells you're going to get per dose. So that's the first thing we learned. It's present in, we think, all tissue culture. It's present in a dose-dependent, they'll be present in a dose-dependent manner, and you can get them through multiple stresses. This was really embarrassing for me. Here's a PC3 cell culture. Here's a giant cell sitting there. 3 days of treatment with high-dose docetaxel. This is what's left in your tissue culture. This is not gradient. This is just put in the poison. I didn't see that for 30 years. Why? Because, well, when we're doing cell culture to see how well a chemo worked, we put in the dye, we count it, or we put it in a hemocytometer and count how many are going by. I never looked. But there they are. And that's what you see. And everybody who's repeating these experiments now, when you actually look through the microscope, that's what you're seeing. Stress-induced giant cells that are polyploid and polyploid. So we said, okay, this is just a tissue culture artifact. Well, turns out it's not. If you do any kind of mouse modeling, xenograft modeling, they're there. Just the pathologists basically ignored them. They said, yeah, that's cancer. Or, yeah, those are cells that are destined to die because that's what multinucleated cells do. So I asked my pathologist, Angelo DiMarzo, do you ever see these? And he said, really, no, except until we stained cell borders with an epithelial marker so we could get the cell borders. In a standard H&E pathology slide, you don't see a cell border, so two nuclei next to each other, everybody just assumes they're two different cells. But when you stain with a cell border, you can start to pick these out. And as it turns out, the more you treat, the more of these appear in the subsequent lesions. They're also present in the peripheral blood of patients with metastatic prostate cancer. My friend Peter Kuhn, who designed an assay to look for circulating tumor cells, had done 10,000 patients of all kinds. And I said, Peter, do you see these? He said, no. The next day, he sent me one of my patients. One of his students did. And I called him and he said, what happened? He said, oh, my computer algorithms don't recognize those as cancer cells. So again, nobody was looking, and they're there when you look. And in fact, now that they've redesigned their algorithms, they're finding them all the time. And they can tell between singles and clusters. It's really remarkable that these rare cells are there. They're there in breast cancer, in the literature. Gonzalo did breast cancer cell lines in our lab and found 2% of MDAs and 3% in MCF7s had these cells. Max Wisha, who uses a filter technique to get CTCs at the University of Michigan for breast cancer patients, said to me, Ken, I see these all the time. We thought they were nothing. But there they are in the blood of breast cancer patients. They're seen in colon cancer. They're in our colon cancer cell lines. CACO2s have 8%. HCT16s have 2. They're there in ovarian cancer. And in this paper published in 2017, they noticed that there were actually more post-chemotherapy in patient biopsies. And in our ovarian cancer cell line, we actually have one cell line that's highly metastatic that's 14% in a stable manner. Reed-Sternberg cells, a multinucleated cell in lymphoma, has been famous for years as a cell that's there, not knowing what we did, but we also see these cells in other lymphomas. They're in glioblastoma, bladder cancer, and we found them in our lung cancer lines. So how are they formed if these are so important, and now we know they're dynamically important? Well, we know they're formed in two ways. From our work of looking at epithelial and mesenchymal cells, we know that they can undergo an aborted cytokinesis. They'll double their DNA, and then they won't divide, and you get them that way. We also know, because we had green and red cells, that they're actually fuse. So this is a PC3 epithelial cell line labeled with a Histone green fluorescent protein. And we put some chemotherapy into the culture and then just followed them. Again, by following these dynamically, we learn new things that we didn't know occurred, like cell fusion. So if this hasn't terrified you enough that these cells exist, seeing cancer cells fuse together to become stronger should scare you. So how are we thinking about how do they survive and beat therapies? So if tumor cell heterogeneity is not the whole answer to drug resistance, how were those giant cells actually surviving in a 10 nanomolar chemotherapy? Well, it turns out that we labeled these with Laura Butita, who's a Drosophila scientist at the University of Michigan, with Fucci labels. And what the Fucci labels do is basically attach a fluorescent protein to different parts of proteins in the cell cycle. So if you, for example, turn on P27 and you put a red marker on that, it tells you a cell's in G0. It's not dividing. So how does chemotherapy kill a cancer cell? So a T cell immunotherapy works when it does, because a T cell recognizes the cancer cell as foreign, punches a hole in it with perforin, and then out comes the cell debris and kills the cell. Radiation will do that if it's strong enough, it'll blow up the cancer cell. But if don't give a lethal dose of radiation, or you give chemotherapy, what you're asking the chemo to do is damage the DNA of the cell, and then when it starts to divide, it recognizes that it's damaged and shunts the cell to apoptosis. So that's why we always say cancer needs dividing cells to work. That's why your hair falls out. Well, not in your case. Your hair falls out, you get diarrhea because it's killing dividing cells. If you don't divide, you can't die. And you're not going to get damaged. So by some mechanism, these giant cells sense stress and go right into G0, which is pretty wild. We also know that somehow they're going to repopulate the tumor. Are they related to those tiny cells, and are they repopulating the tumor? Well, the paradigm for that is cancer stem cells. Cancer stem cells are known to asymmetrically divide to repopulate a tumor. Max Wisha actually discovered solid tumor cancer stem cells in breast cancer several decades ago, and he helped us say, well, do these have stem cell properties? We took the big cells, compared them to their 2N cells that were also in the population, and they actually have many canonical stem cell markers. We haven't proven beyond a doubt that they're stem cells, but they do express KLF4, c-MYC, NANOG4, SOX2. And so we think that these have the ability, once they come out of hibernation, to undergo asymmetric division. And in fact, we've seen not only symmetric division, we've also seen where we think that those little cells came from by capturing an event like this. So this is a giant cell that's been actually caused by the fusion of an epithelial and a mesenchymal cell. I didn't show you the markers, but there's red and green markers there. We actually caught this one budding off a tiny cell. This is not asymmetric division. This is a process called meiosis, which is controversial in itself because, again, you have to catch these events live with movies because people have seen this, but they go, we don't know what that is because it's static. So, but we think that these are transient amplifying stem cells that then repopulate that tumor, that bloom you saw. And in fact, again, if you look at, in this case, LNCAP cells and treat them with docetaxel, another form of prostate cancer cells, 90% of the surviving cells are these polyploid cells, and then wait 3 weeks later, they're back to 3% exactly. So there's some kind of sensor in the population that says we need 3% of these giant cells. So how do you explain that? And it goes back to 3% when you've repopulated? I mean, that's another, we don't get that at all. And why? How do it know? How does it sense that? How do you explain that evolutionarily or ecologically? And not only that, when those 2N cells come back, they're not only resistant to the therapy you treated with, they're resistant to other chemotherapies and radiation therapies, which is what the far graph is showing you, that we've had sensitive cells that we then exposed to radiation, that these cells versus these cells are resistant to radiation after being treated with chemo. So we think they're stem cells, but we also have this meiosis process going. You don't need to be STEMI to do that, and it's all highly controversial. So how do we put all this together? Well, we think that the first of these of these giant cells, may be formed back in the tumor microenvironment in the ecosystem of the cancer, primary cancer, because all you need is one, and all you need is that one to potentially escape into the circulation. It's already in a dormant or hibernating G0 state, so it can avoid the stress of circulation. And in the case of prostate cancer, it can go to bone and sit there in a lag period or a dormant period for years until it grows up into a clinical metastasis that we can see, which we then treat. You have this you get rid of all the 2N cells, but then these cells come back and then reformulate your metastasis, and the whole time you can be spreading to other sites. So this is what we think is potentially going on. As a sort of a floater idea, what we've thought about is calling these keystone cells. After keystone species. Dr. Karina came up with this term to think about cancer stem cells in a paper she published. But keystone species, as I'm sure everybody in this room knows, are species like the wolves of Yellowstone or the elephants of the Serengeti, where these species are small in number, but have huge ecological impact when you take them out. So if we think these are few in number, and if we could target them, potentially the whole paradigm of the ecosystem would collapse. And so we think so it's a good way for us to explain it, but it's a very messy way to think about it for ecology and evolution. Because is it a new species? Is it a different species? How do you explain this? And also, how do you explain again this idea that they're there, maintained in the population until you stress the population, then they take over and say, okay, we're going to survive that stress, and then we're going to repopulate. So we're looking for new ways to understand that and describe it for ourselves. And so we thought about, the queen bee in the hive, but we also thought about this keystone species idea. And actually, this is what we've been calling them of late. We've come up with several different ways that we're thinking of targeting these, which I'm really not going to talk about today, but we're going to hear about P53 as a potential. There's a kinesin called kinesin 14 or KIFC1, which is actually used for, the cells use KIFC1 to asymmetrically divide. Something this ugly should have a specific immunoprofile that may lead it to be subject to something like immunotherapy. But my guess is they've downregulated their MHC complex and they're silent to, will not be recognized by immunotherapy. But in this day and age, we have to look. And of course, if we could deliver alpha particles, alpha radiation, to these cells, that would kill them too. Because again, you don't need dividing cells if your dose of radiation is high enough. So those are several things we've been pursuing. And I'm going to end there acknowledging my old group, especially Sarah, who's been leading much of this effort and is really key to moving this forward with me. Lori Kosteka is a PhD grad student who has now come into the lab and is working on this. And Gonzalo has moved on, but he was very key in this. And I thank you for your attention.
Hi, thanks. It's a nice talk. So I have two questions. I was wondering if you had taken any of these giant cells and transplanted them, into mice to see if they have more of an advantage to grow than other cells. Is there any way you can sort for those cells through flow cytometry and then?
Yeah, so great questions. And I've shown you all the data we have, but we're working very hard. Lori has just figured out in literally the last month a way for us to sort these cells.
Yeah.
The problem is when we make them, which we can make, now we know we can just pour some chemo on and make them, but as soon as we take the stress away, they repopulate the 2N. They don't stay the polyploid cells, so we can't grow up a population of polyploid cells. With Max, we have planned to do tumor-initiating experiments where we'll do serial dilution, put one in the back of a mouse, 10, 100, to show that they're stem cells. But again, the key, what we have to keep in mind constantly is that these don't sit there as giant cells if there's no stress. As soon as whatever mechanism they use recognizes no stress, they start to pop out the 2N cells and become a minority population again, which has challenged us to study them at all until Lori figured out how to filter them and grab them. And we can flow for them, do FACS sorting, but that just does so much damage to the cells and takes so much time. When there's such a minority population, we haven't been able to do that.
And also, your statement about the cancer drivers, I just thought that was interesting because I work on mouse models, and one of the issues we have is that the mouse models in the lung cancer field have led to groundbreaking findings, but they are simplistic in terms of they don't have a lot of heterogeneity, but there are oncogenic mutations, tumor suppressor loss that do drive the cancer. So in terms of saying that there's no cancer drivers, how would you sort of address that in terms of, there's certain mutations that we can induce that don't lead to tumorigenesis or growth or, it's an interesting point.
Yeah, so there are mutations that are very important in cancer. Sure. RAS, P53, MYC, et cetera, that would be considered classic drivers. Yeah. But those aren't enough to explain heterogeneity or resistance.
Exactly. Yeah.
And in fact, even in using the BCR-ABL paradigm, Gleevec didn't cure most people. They get resistant, right? So and then for most solid tumors, true drivers have not been identified. But every mouse model is pretty much based on drivers, right? And they're important.
Which is an issue.
We need to understand them, but they don't explain, and one of the reasons you can cure a mouse is it doesn't have that heterogeneity.
Well, not yet.
Right, not yet. But, well, hopefully you'll do that for us. Yeah.
What's the relationship between the multinucleated cells you see in cancer and those that exist in healthy tissues in the body?
Yeah, so the question is, what's the relationship between cells and the normal cells in the body that do this? For example, macrophages are very good at becoming multinucleated histiocyte in response to chronic inflammation. Osteoclasts do this every day to help remodel bone as multinucleated cells. So we think, that again, cancer is just using mechanisms that have already been in place through normal biology. You have to have the right receptors to fuse, et cetera, et cetera. So it's just using what those cells do normally to combine and make more genetic material. But we think those mechanisms are important and can help us, learn what's happening here. But that's basically all I've got. You want to follow up?
The heritability thing, you see the single nucleotide off. What's your thought about how the heritability of whatever it is?
Yeah, we want to know, and we don't know. And one of the things that one of the huge mysteries here is, okay, I understand how a giant cell can be resistant because it's basically not dividing. But how is it using that time where it's exposed to the stress to learn, quote unquote, and I do say that in quotations, to pass off resistance to the smaller 2N cells? And what are the mechanisms? There's got to be multiple mechanisms to do that. We know there's multiple mechanisms of drug resistance, but how it passes that off, we want to study that. We don't know. Katerina?
So can you control the proportion of the polyploid cells? I mean, if you increase the amount of treatment, you get higher proportion. If you decrease it, you get a lower one. Can you keep it under control?
Yeah. So the question is can you change the ratio? And so far, but we've only been doing this for a few months. So far, in vitro, in tissue culture, we have not been able to change that percent. Like PC3, it's 3%. Hit it with this chemo, hit it with that, it's 3%.
But you change the amount as well.
Yeah, but well, they change what?
The intensity of it.
Yeah, but they just go back to that set point. Now, somehow, in like SCOV cells, they're 10, 14%. I don't know how that happens. We actually have an experiment going right now where we're basically sampling every day over the space of 21 days, counting the number of giant cells versus 2N cells to try and understand some of those dynamics and get some of the math, how are they formed in that. But so far, we know that in vitro we can't do that, but I also know that in vivo, in a patient, we know that the more the patient's been treated, if you look at their metastatic lesions, the more of these cells are in the metastatic lesion. So primary, it's very difficult to find them at all. But in a guy who died of prostate cancer who had, and you look in their metastases, you can find several of them in a field. So in patients, there's more of them, but we have not been able to recapitulate that.
You have used like the maximal tolerable dose almost.
Have we,
There you have been using maximal tolerable dose.
yeah. Mm-hmm.
Very interesting that the population keeps rebounding to 3%. And
Yeah.
I think you mentioned in the end potential strategies to get rid of those cells before chemotherapy. And I was wondering if you have thought about exploiting the fact that these cells require more energy basically to grow. Might they be more sensitive to starvation or other sort of nutrient?
Yeah, the energy requirement of these cells is clearly different by look, but I have no idea. We have not had enough time to study that, and doing metabolism type experiments, like using a seahorse or something, is going to be really difficult because we can't create a static population of these to study. So, but if you want some to study, we're happy to give them to you or tell you how to make them, because that's not, we just don't know how to do that. There was, yeah.
For an aneuploidy or polyploidy is not always a bad prognostic sign in cancer. In hematologic malignancies, sometimes it has no influence on prognosis.
Yeah. So we have, that very true, that the comment was that aneuploidy is not, in liquid tumors like leukemias, is not always a bad prognosis. We've only looked in the solid tumors so far. We have not tried to move this, understand this in terms of leukemia. And, again, we have to differentiate these cells, this hidden population of cells, from general aneuploidy, because PC3 cells are aneuploid. Every one of them is aneuploid. And how these are, if you study ploidy globally or gene expression globally, these are just buried as such a small percent, you're never going to see them. So I don't understand. One of the things we want to know is how does ploidy play into this? And again, we're working on how do you isolate enough of these to study them as a group or as single cells so that we can understand the relationship to the greater aneuploid population. In fact, we've had, as an aside, we've had trouble describing these because like cancer cells, they're aneuploid. So they're not 2N, they're 2N plus or 2N minus, there's something else. So we can't just say these are 4N or 8N, they're aneuploidy of aneuploidy. Yeah. Mm-hmm.
I was wondering, did you ever look for signs of somatic recombination that are induced in these big cells so that you basically have this as their way of sexual reproduction?
Great idea. We have not. We haven't had time, but it's a great idea.
In one of the initial videos you showed us with the hexagons, I remember you showed us like the polyploid cells, and there were some small ones that were kind of like following after. Have you investigated what those may be?
Yeah, so we think those are we think those are a product of this meiosis that they're budding off of the big ones, based on and that they are the only way I can explain it is that they're transient amplifying stem cells on their own and repopulating, but we have to prove that.